this format. Okay. I think you guys should all be able to hear me now, and someone should give me a holler if not. But it's really a pleasure to be here today, and thanks to Don and to CDH International for inviting me. My name is Frances High. I'm a clinical geneticist with the CDH Genetic Study at Massachusetts General Hospital and Boston Children's Hospital. And I'm going to talk today a little bit about some of the research updates from our study. I'll tell you a little bit about who we are, what our goals are, and I'll tell you a story of a new gene that we've been studying that hasn't been published yet, so you guys get to get a little preview. So this is our team. We are a group of very diverse skill set, I would say. So we are geneticists, we are surgeons, pediatric surgeons. We have bioinformaticists who help us understand and interpret our genetic data. And we are bench scientists as well, people who do experiments in the laboratory to try and understand the function of CDH genes as we uncover them. There are a number of people that I should give a shout out to. Most of them are in this photograph. In particular, I need to acknowledge Dr. Pat Donahoe, who really initiated this project. She's a pediatric surgeon and is very passionate about CDH and initiated this project now almost 2 decades ago. And I've been on board for about the past 10 years, but this was really something that she started, and then the members of the laboratory that I've noted here. We do not work in isolation. We're a part of a very large group of collaborations, and I've just pointed out here on this slide some of the really important people. So we work closely with the team across town at Boston Children's Hospital and have for the entirety of our study. Jennifer Liu is the study coordinator, and she is part of today's meeting and is floating around in the chats and is definitely available for contact and questions. And Dr. Jill Zalikis is a pediatric surgeon over at Boston Children's Hospital, who's the PI for the study over there. We are fortunate to be funded by the NIH as part of a large multi-center program project grant, and we, for almost the past 5 years, have been collaborating directly with Columbia University and the Care for Kids Dreams study. So I'm very pleased to be following Julia, who gave you a nice overview of that study and the really terrific work that we've been able to do together. We also have, as part of our work, collaborations with the University of California, San Diego, and the Jackson Laboratory, both of which are doing really fabulous work to try to uncover the mechanisms behind some of the CDH genes that we've been discovering. And then last but not least, of course, we are tremendously grateful to the patients and the families who participate in the study and who really advocate for the importance of research on this topic. So our CDH study, I was counting, has actually been ongoing for 18 years and is going strong. I believe that I'm not mistaken in saying that this is the oldest or most long-standing CDH genetics study in North America. We have to date recruited approximately 700 individuals with CDH and about 1,300 unaffected family members. So to give you a little bit of a sense of what we do as part of the study, we use extracted DNA for genomic studies. It's very similar to what Julia was talking about with DREAMS. We also, whenever possible, like to obtain a skin biopsy sample from the time of the surgery or blood that we can actually make cell lines in, and we can then propagate those cells in the laboratory and do functional studies to try and understand how the genetic changes that we identify actually affect the way in which cells or cell types that are important for lung and diaphragm development behave. We are also beginning to do some really interesting and I think innovative work using cells that we're directly collecting from tracheal aspirate samples in newborns with CDH. So for those of you who have had a child with CDH who's been in the intensive care unit and was intubated, you know that as part of their routine clinical care, there are occasionally suctions just to kind of get all that fluid out of the lungs and allow the endotracheal tube to do its job. So what we're actually doing is taking that fluid that would have traditionally been discarded and actually isolating cells from that and propagating them in the laboratory. And it appears that these cells actually tell us quite a bit about the biology of the lungs in these infants. And so we're just beginning to do that over the course of the past year, and we're interested in seeing how what we can learn about lungs from CDH babies using that technique. As part of our study, we are also doing detailed clinical phenotyping for disease severity and comorbidities, such as additional birth defects, because we're, of course, very interested in understanding which specific genetic defects are associated with better or worse clinical outcomes, as well as additional health concerns. So as I mentioned, we've recruited about 700 individuals, but through our collaboration with DREAMS, we've been able to increase our total cohort size to greater than 2,000 patients. So when we talk, I think, Julia gave a nice overview and of what we know and how much we don't know about the causes of CDH. And I like to use this analogy of the tip of the iceberg. So our current understanding of the genetic causes of CDH is really growing. We know that there are single gene disorders that are syndromic, meaning they cause other health concerns in addition to CDH. We know that some of those are associated with CDH. We know that certain chromosomal disorders are associated with CDH. And through all of the studies we've been doing over the past decade or so, we are gradually uncovering more and more genes that, when affected, can cause CDH. And so I've just mentioned a few of these here. There are inherited genes, may not affect everyone in the individual. This includes this example that Julia just gave in the last talk of GATA4, but there are other examples similar to that. And then there are also genetic changes that are de novo or new in the patient with CDH that are not inherited from the parents. And we think that that accounts for about 20% of CDH cases. But then there's the whole rest of the iceberg under the water. And so there's a lot that we don't know and a lot that is still to be discovered. And so we're using multiple different techniques to try and understand what's under the water. And so really the overarching goal of our research is to take what we can learn from human CDH genetics, also from mouse CDH models, and also from laboratories on CDH genes. As I mentioned, we do a lot of cell culture-based work, for example, to try and understand the function of these genes. And if we put all three of these different techniques together, we begin to sort of see patterns. So there are common pathways and developmental mechanisms that can underlie CDH, even though there may be hundreds of genes that contribute. And our goal is that ultimately these, we can take what we learn from these common pathways.
Realistic, right? A little bit older.
I don't know if maybe people need to mute. I'm hearing some background noise from other individuals. Right. Sorry. But our goals here are, we have two major goals. First of all, we would like to improve genetic diagnostic and prognostic tools. So a lot of people come in and, we don't have a lot of great information we can give to families with CDH about, about whether genetic testing can give us an indication of whether there be other health concerns that individual might have, and also whether there might be a risk for them to have another child with CDH, for example. And then the second goal will be to hopefully identify therapeutics, novel therapeutics that are directed at lung development. So I just want to spend the rest of the time talking about one example of a CDH gene that was identified through a global collaboration. And I've just put a bunch of names on the slide because this sort of gives you a sense of how many people have to work together to just describe a single gene. And so this story comes from several individuals in the United States, as well as a collaborator in France. This is a story about a form of CDH that is caused by mutations in this gene called PLS3. So Julia described a little bit about what a pedigree is. So this is how we draw families. And again, the squares are males and the circles are females. And so you can see that here is this family one is a very large family. And the males in this family were affected with CDH. Similarly, families through 6 have affected males. And when we sequenced all of these families, we were able to identify changes in this gene PLS3 that were present in the males that had CDH and also in the females, their mothers basically and sisters that were carriers. So remembering again from basic biology that females have 2 X chromosomes and males have 1 X and 1 Y chromosome. And so this is the reason why X-linked disorders such as this can result in symptoms in a male, but not in a female, because the female has an extra kind of backup copy of this gene. So the other interesting thing about this gene was many of the individuals, in addition to having a diaphragm defect, also had another type of hernia, which is a hernia of the anterior abdominal wall. And so this on the left is an image of a woman who has a scar above her belly button. And this is because she had a hernia there above her belly button that needed to be fixed. On the right, this is actually an ultrasound image. And what you can typically see if you look at anyone's abdomen is you would see what's on the top here, which is 3 different layers. These are the muscles of our abdominal wall. These are the things that help us do sit-ups, basically. In this individual who had CDH and a PLS3 mutation on the bottom, you can see that instead of there being 3 muscle layers, there's only 1. So there was somehow missing or improper development of these other muscle layers in the body wall. So this seemed to be a pretty specific finding in these individuals. So in the 6 families that we described, they all had diaphragm defects, and 3 of the 6 families also had these anterior abdominal wall defects. I'm just showing you here the details on the genetic information, the genetic findings in these families. The details are not particularly important to understand, but I think it's relevant to think a little bit about how we try to understand genetic information when we do these sorts of studies. So there are a few things that we can do to try and understand whether we think a genetic change is meaningful or not. Because of course we all have many genetic changes, and that's what makes us unique individuals, right? So the first thing that we often do as geneticists is we look in large population databases to understand whether or not that specific genetic change is found in healthy individuals. And so geneticists use this database that's called NOMAD, and it contains over 100,000 individuals that presumably do not have CDH. And what we found is that all 6 of these changes that were found in the CDH families were not seen in this very large population database. The second thing we can do is we can try and predict whether the change in the gene might result in a change in the function of the protein. And so we can do that using these sort of computational scores like this CAD score. But the other thing that's really interesting to do is we can look at the region in the protein, and we can try and understand whether or not that is conserved or the same across different species. So if we look at the parts of the genes that are disrupted in these 6 families, and we look at all these different vertebrates, rat, human, cow, mouse, hamster, zebrafish, we can see that that part of the protein is conserved, is the same, even all the way down to yeast. So that gives us confidence that this is a really important part of the protein. So what is this gene? What does it do, and how could it potentially be involved in CDH? And this is a question we're still addressing. So it's a really interesting gene. It actually it does something really fundamentally important in all cells, and that is that it binds the actin cytoskeleton. So the actin cytoskeleton you can kind of think of as being like a scaffold for how our cells change shape and do all sorts of important functions. And what PLS3 does is it actually binds together these different actin things, and so it's been shown to be important in things like cell migration, cell morphology changes, and other really fundamental processes in biology. So we decided to look and see what the CDH-associated mutations in PLS3 do to cells when we grow them in culture. And we showed that, that in these 4 panels on the right here, that if you have these CDH-associated mutations in the cells, these cells really have fundamentally different shapes. And you can see that they have, you might be able to appreciate that they have many more of these projections coming out of them. And so these are things called filopodia and lamellipodia, and these are really important for how cells migrate. So we think that these PLS3 mutations that are associated with CDH are playing some role in the actin cytoskeleton and how these cells behave. We can also look to see where this gene is expressed developmentally, where it's turned on developmentally. And so this is just an image here. We turn to the mouse embryo to try and understand this. So this is a little tiny mouse embryo, and blue is just a depiction of where this gene is turned on in the mouse embryo. And we're looking at this time point, which is equivalent to when the diaphragm is forming in the mouse. In the bottom panels here, the L is the liver and H is the heart. And sandwiched in between the developing liver and the developing heart is the diaphragm, and you can see that it's blue here. So we know that this gene is turned on in the developing diaphragm. We also know that the gene is turned on in the lungs, and you can see that in the staining in brown on the right, and this is actually a human lung specimen. And you can see it in both the mouse and the human. So we went on to model this gene in mouse in collaboration with the Jackson Labs in Bar Harbor, Maine. And so I need to acknowledge Carol Bolt, Rick Mazer, and Julie Wells for this work. And what we found is that these mice have diaphragm defects. They have holes in their diaphragm. And so that's depicted up here. These are just dissections showing a mouse diaphragm. And so some of them have holes in the diaphragm, and some of them don't actually have holes, but they have thinning of the diaphragm, either in the anterior portion here, the red arrow, or the posterolateral region with the green arrow. And the other interesting thing about these mice is that they also have these anterior body wall defects as well, which again mimics what we saw in the human patients. So the final thing I want to mention about this gene is that it was a little puzzling to us because this is a well-known gene in human genetics because the gene has also been shown to cause osteoporosis and bone fractures. And this is just this was a study of how when this was discovered back in 2013. And so these patients do not have CDH, but they do have very fragile bones and can break them easily. And so we went and looked in both our human patients as well as our patients with our mouse model to see what the effect is of these specific mutations in PLS3 on bone density. And what we see is that in one human patient, this patient actually had increased bone mineral density. So basically, you could think of it as being like stronger bones compared to the average. And similarly, we saw exactly the same thing in the mouse model, and that's just depicted by these graphs here. So overall, you know, our conclusions were that really it depends on which type of mutation you have in PLS3. Some mutations in PLS3 can cause osteoporosis, and some mutations in PLS3 can cause CDH and seem to actually increase the density of your bones. And we're really still trying to, you know, to tease apart why exactly that is. What is the difference between these different mutation types? So I just want to conclude now by saying a few important points. I think, first of all, CDH genetics are complex. We have learned over the years that forming large consortia for the study of CDH genetics has been proven to be a powerful tool, and that global collaboration among CDH researchers is essential. We think that by combining human genetic studies, mouse modeling, and other laboratory experiments, we can gain a lot of insight into CDH, and that better understanding of the genes will form a base for better diagnostic tools and ultimately transformative therapies. I've also shown you this novel gene, PLS3, that causes an X-linked familial form of CDH. And I also shared this the story of PLS3 with you because I think that it illustrates a point that we come across all the time when we do rare disease research. And that is that in the process of studying a gene that causes CDH, we've actually kind of stumbled across or learned something really interesting about bone biology and probably some insight into osteoporosis, which is, of course, a tremendously common public health concern. So, you know, when we think about advocating for research for rare disease, we're all here because we're really passionate about CDH, and we'll continue to be really passionate about CDH. But I think that it can be helpful in advocating for CDH research to remind people that when you study a rare disease, you learn a lot of really interesting things about basic biology, and that can provide insights into many more common diseases and have very broad impacts beyond our community. And with that, I just want to make sure that you have our contact information. If you're interested in participating in our study, you can contact me directly. You can also contact Jennifer Liu. This is her email address here. We do have an exhibit booth, as Don has mentioned, and should have all of our contact information. Please feel free to give me a buzz. I'm happy to talk about anything. Thanks so much for having me.